In this video, I will explain the chemistry of phospholipid in detail. So, phospholipid, it is a type of compound lipid, and this phospholipid contains phosphoric acid as the additional group. And these phospholipids are of two types on the basis of the type of alcohol group it has, glycerol or sphingosine. So, glycerophospholipid having glycerol as the alcohol group, and sphingophospholipid having sphingosine as the alcohol group. So phospholipids are of two types, glycerophospholipid which contain glycerol as the alcohol part, and sphingophospholipid which has sphingosine. Coming to glycerophospholipid, so the alcohol group is glycerol, and the basic structural unit for a glycerophospholipid is the phosphatidic acid. So this is the structure of phosphatidic acid. This is glycerol, CH2OH, CHOH, CH2OH, and the first and second hydroxyl group, they are esterified with fatty acid, and the third hydroxyl group is esterified with the phosphoric acid, and this is the basic requirement for a glycerophospholipid, and this basic structural unit is known as phosphatidic acid. So this is the structure of phosphatidic acid. So in phosphatidic acid, the structure contains one glycerol molecule and two fatty acid, and the third hydroxyl group is esterified with the phosphoric acid. And this phosphoric acid is now attached with a base, and that base can be a nitrogenous base or non-nitrogenous base. So on the basis of the type of base, these glycerophospholipids are again subclassified into nitrogenous glycerophospholipid or non-nitrogenous glycerophospholipids. So glycerophospholipid structure has four components. One is glycerol molecule, and the first and second hydroxyl group esterified with the fatty acid, and the third hydroxyl group is esterified with the phosphoric acid, and that phosphoric acid is now attached with a base. Then that base can be a nitrogenous base or a non-nitrogenous base. So these are the examples for nitrogenous bases: choline, ethanolamine, and serine. And inositol is the example for non-nitrogenous bases. So glycerophospholipids can be nitrogenous glycerophospholipids or non-nitrogenous glycerophospholipids. So these are the examples for nitrogenous glycerophospholipids: phosphatidyl choline, which contain choline as the nitrogenous base; phosphatidyl ethanolamine, which contain ethanolamine; and phosphatidyl serine contain serine as the nitrogenous bases. And coming to non-nitrogenous glycerophospholipid, phosphatidyl inositol and cardiolipin. These are the examples for non-nitrogenous glycerophospholipids. Coming to phosphatidyl choline, and this phosphatidyl choline is also known as lecithin. So phosphatidyl choline is a nitrogenous glycerophospholipid which contain choline as the nitrogenous base. So this is the basic structure of lecithin: phosphatidic acid with choline. Then you should know the significance of lecithin. And this lecithin is an important component of lung surfactant, and the normal lung function depends on the supply of the lung surfactant, and these lung surfactants are produced by the type 2 alveolar epithelial cell. So what is their function? This lung surfactant helps to reduce surface tension of the alveoli, thereby they helps to prevent lung collapse. So there are two types of alveolar epithelial cell, type 1 and type 2, and this type 1 alveolar epithelial cell, they are concerned with the exchange of respiratory gases, and type 2 alveolar epithelial cells, that is concerned with the production of lung surfactant. Coming to the deficiency manifestation of lecithin, that is respiratory distress syndrome. So respiratory distress syndrome is due to the deficiency of lecithin. It is the major lung surfactant, and which can result in respiratory failure. So you should know what is LS ratio, lecithin sphingomyelin ratio of amniotic fluid. So it is an index of fetal lung maturity. So during the early stage of pregnancy, there will be more concentration of sphingomyelin. So towards the end of the pregnancy, there will be more concentration of lecithin. So ratio of 2 indicate full lung maturity. Coming to cephalin. Cephalin is also known as phosphatidyl ethanolamine, where the nitrogenous base is ethanolamine. So cephalin is phosphatidic acid plus ethanolamine. Coming to phosphatidylserine. Here the nitrogenous base is serine. So phosphatidylserine is phosphatidic acid with serine. Coming to phosphatidyl inositol. Here inositol is a non-nitrogenous base. So phosphatidic acid with inositol is phosphatidyl inositol. And usually this phosphatidyl inositol is phosphorylated. It's a phosphorylated compound. So phosphatidyl inositol on hydrolysis produces diacylglycerol and inositol triphosphate. So what is the significance of this diacylglycerol and inositol triphosphate? This diacylglycerol and inositol triphosphate act as a second messengers for hormonal action. So there are certain hormones, they have receptor on the cell membrane, and these types of hormone require second messengers. And diacylglycerol and inositol triphosphate, they can act as second messengers for mediating hormonal actions. And cardiolipin is also an example for non-nitrogenous glycerophospholipid. This is the structure of cardiolipin. It is made of two molecules of phosphatidic acid and it is connected by one molecule of glycerol. That's why it is known as diphosphatidyl glycerol. Two molecules of phosphatidic acid and one molecule of glycerol, diphosphatidyl glycerol. And first it was isolated from the heart muscle and it is present in the inner mitochondrial membrane. What is the significance of this cardiolipin? This cardiolipin has antigenic properties. So cardiolipin is the phospholipid which has antigenic properties. Coming to the clinical significance of this cardiolipin. There is one clinical condition known as APLA syndrome, that is antiphospholipid antibody syndrome. It's an autoimmune disease where antibodies are produced against cardiolipin. And in pregnancy, this APLA syndrome can result in recurrent abortion and preterm labor. So this is the classification of phospholipid. So we can classify phospholipid into glycerophospholipid and sphingophospholipid. And this glycerophospholipid again classified into nitrogenous glycerophospholipid or no nitrogenous glycerophospholipid. The examples for nitrogenous glycerophospholipids are phosphatidyl choline, phosphatidyl ethanolamine and phosphatidyl and phosphatidyl inositol and cardiolipin. These are the examples for non-nitrogenous glycerophospholipid. There is another topic, phospholipases. So phospholipases are enzymes which can hydrolyze phospholipids. One of the type of phospholipase is phospholipase A2, and this phospholipase A2 act on lecithin, that is phosphatidyl choline, and on hydrolysis this lecithin produces lysolecithin, and this lysolecithin is a powerful hemolytic agent. And that enzyme phospholipase A2 enzyme is present in some snake venom. That's why snake venom can produce as hemolysis. So that's all regarding glycerophospholipid. Thank you.